Welcome back to Human Biology with Dr. B. Today we're talking about the neuron. So we have two major cells of the nervous system. The first one is a neuron, and these are what we refer to commonly as nerve cells. They are excitable and they transmit messages. Three main types: sensory, motor, and interneurons. Neuroglial cells, these are supportive cells. They help cells not necessarily regenerate, but to keep the cells healthy, to allow them to continue working. Because as we remember from our discussion of both muscle and neuron cells, that neither of these truly go through mitosis. They spend most of their time in G0 just acting like muscle and nerve cells. And so with muscles, we have satellite cells to help repair and replace and to regenerate. Well, as here in the nervous system, we have neuroglial cells. So we have three different types of neurons, as we talked about. The first one is a sensory neuron. Now, sensory neurons bring information towards the central nervous system, the central nervous system being the brain and spinal cord. Motor neurons carry information out, away from the central nervous system, and this usually goes to a gland or an effector. The general term is effector. It can either be a gland or a muscle cell. And then we have interneurons, and interneurons is where all decisions are made in the central nervous system. Interneurons take the information coming in, they process it, they bounce it around different areas of the brain so that the body can make decisions as to what to do. These are only in the brain and spinal cord. They're between sensory and motor neurons. They both interpret and integrate. Integration is really important here, sensory information. And even though we have a ton of neurons reaching all of our muscles and all of our glands and all of the organs in our body, interneurons in our brain and spinal cord account for 99% of the body's neurons. Kind of amazing. That's how big your brain is. Now, the structure of a neuron, it's a stellate shape, and this allows for communication with other cells. And there are three main parts. We have the dendrite, the axon, and the cell body. So follow me here. Here are the dendrites. The dendrites, their job is to receive information from other neurons or from the environment. Dendrites can be any type of our sensory receptors. So if you were to pinch your finger, you would be triggering your dendrites to send a message to your brain. The cell body is basically where all of the typical cell parts, if you will, the organelles reside. That's where we have the nucleus, we have the endoplasmic reticulum, where the neurotransmitters are made. This is also where the input is integrated. Then we have this very long axon, and the axon conducts information away. Axon, away from the cell body, either to the brain or spinal cord or to another neuron. Now, in your body, you have axons reaching from your spinal cord all the way down to your muscles and then some back. It's pretty amazing. We'll talk more about that when we talk specifically about the central and peripheral nervous systems in the next lecture. So nerves. Now, nerve is the layman's term that we use for neurons. Now, neurons are the cells, the single individual cells. A nerve is parallel axons, dendrites, or other neurons of any type moving in one direction. These are covered in tough connective tissue, and they're typically classified as either sensory nerves, motor nerves, or mixed. And mixed nerves have both sensory and motor neurons associated with them. It has a very similar structure to a muscle. So if you think about the muscle and the muscle fiber right here, and where there's a number of muscle fibers in a fascicle, and the number of fascicles make a muscle, it's the same way with a nerve. We have neurons right here, and they are grouped together in sensory or motor, and then from there, they are grouped together with more connective tissue in what we call a nerve. And this is so that, because if you think about all the muscles in your lower leg, and if you've ever had sciatica, you know what can happen when anything presses on that neuron or on that nerve. You feel it throughout all of the muscles of your body. Now, these neurons are packaged together so that they're less easy to separate, if that makes any sense. So they're more protected as they move through the muscles, around the muscles, through the bones, and around the bones. Now, the structure of neurons looks something like this. They have a myelin sheath. Now, the myelin sheath is basically, if you think about a cord, so you're on your computer right now looking at this video, and in some cases, your computer is attached to an electric cord so that you can charge it and so that it doesn't die on you in the middle of the lecture and then you miss half the lecture and you're like, oh no. The same thing is with the myelin sheath. If you look at the electrical cord attached to your computer, it's not just empty wires. If you touch the electrical cord while it's plugged in, you're not going to get zapped. And the reason why is because it has this rubberized protective coating around it. The same thing for the myelin sheath. This is found on most axons outside of the central nervous system, and it provides electrical insulation. Now, the myelin sheath in the peripheral nervous system is everything outside of the brain and spinal cord. The Schwann cells, because Dr. Schwann discovered Schwann cells, form the myelin sheath. And what they do when these myelin sheath is being formed is the Schwann cell wraps in around itself to protect the axon. Now, if you'll look up here at this picture, you'll notice that the Schwann cells are not like the electrical coating, the rubberized coating of the electrical cord on your laptop. There are gaps between them, and we call these gaps nodes of Ranvier. And the myelin sheath allows the electrical impulse to move much quicker than it would if it had to travel the entire length of the axon. Because the electrical impulse basically, right here, this is called the axon hillock. This is where the action potential starts. We'll talk about the action potential in just a second. So right here, and then the electrical impulse starts here and moves. It jumps from one to the next. And we call this saltatory conduction. Because honestly, a salt, if you think about it, if you think about table salt, sodium chloride, and in this instance, we're moving sodium and potassium. It's still salt. And so the electrical impulse just jumps. And that allows faster communication down the axon. So the neuronal impulse, we refer to this as an action potential. The action potential is an electrochemical signal. It's sodium potassium pumps, and they basically change sides of the membranes to allow for the electrical impulse to carry the entire length of the axon. Now, within the axon, there are specific ion channels, and there are sodium ion channels and there's potassium ion channels, and they are specific for either ion. Ions are also moved by the sodium-potassium pump. And if you remember back when we were talking about the cell, the sodium-potassium pump is one of the best examples of active transport. And remember back to the definition of active transport, it takes energy to move the ions against their gradient. Now, the sodium-potassium pump is incredibly important because this is what generates the gradient to allow the electrical impulse down the axon to occur. Now, the sodium-potassium pump pumps sodium ions out of the cell and potassium in. Keep that in mind. And it takes energy. So if you think about when you take an exam, how you kind of feel tired at the end and maybe a little hungry, maybe a little angry, it's because honestly your sodium-potassium pumps are working overtime, chewing up energy, and all of a sudden you need to eat some more in order to put it back into your blood and into your brain cells. So the action potential looks like this. When the neuron is stimulated, there is a reversal of charge across the membrane. So when the neuron is at rest, the plasma membrane inside is negative. So the sodium is pumped out, potassium is pumped in. Now what happens is the loss of the charge difference occurs as the sodium ions enter and allow for a more positive charge in the inside of the membrane. So if you look here, and it occurs in a wave. So sodium gates open, sodium comes in, potassium rushes out. Now, in order for an action potential to occur, a minimum charge needs to be able to cause these sodium gates to open. And when this happens, this triggers what we call depolarization. And this is a reduction of the charge across the membrane throughout the entire axon. And this occurs in a wave pattern, as you can see here. So here we see potassium getting pushed in or pushed out, and potassium rushing in, and this occurs in a wave down the axon. Now, repolarization is reestablishing the charge difference across the membrane. Now, sodium out, potassium in. Sodium, Na+, so we pump 3 sodiums out for 2 potassiums in. Now, potassium also has a charge of +1 in an ionic form. So when we pump 3 sodiums out for every 2 potassiums in, that means that the net charge across the membrane is going to be negative on the inside because not as many positive charges are getting pushed in. And then when these channels open up, it allows for diffusion, for the sodium to rush in down its concentration gradient, and there's no potassium on the outside, so the potassium rushes out, and this is what causes the electrical impulse and allows you to be able to process the words that I am saying and be able to hear, and your brain to process that. It's all in action potential. It's kind of amazing. This is what it looks like. So here, resting potential is about at -65 millivolts. And so threshold is right here. Threshold just slightly higher. And what happens is here we get a stimulus. We get a stimulus right about here. It allows threshold to be reached, and immediately, it happens immediately. The action potential occurs. Sodium ions rush in, potassium ions leave. So we come down, we hyperpolarize a slight amount, then we come back up to resting potential, and we're ready for another action potential. Now, action potentials are all or nothing. They either go or they don't. Now, that sounds kind of like duh, but you have to understand we must reach the threshold. We must have that much of a depolarization in order to trigger the action potential to send the electrical impulse the entire length of the axon. So once it's started, the action potential does not diminish. So what does that mean? It is not like the waves in the ocean where they start out big and then by the time they hit you on your feet, they're little tiny waves. No, the wave goes all the way through without losing any power, any of its electrical charge. It does not vary in intensity with the strength of the stimulus. The thing that affects how much you feel it or how great it is perceived is how many stimuli, how many neurons are stimulated. And then we have a refractory period. So one neuron can be stimulated multiple times, but there is a very short, very quick refractory period where the neuron cannot be stimulated again while it repolarizes. Now guys, this happens without you even noticing it. You are hearing my voice and immediately, your brain is immediately processing it. So it's not even noticeable the amount of time that it takes these neurons to depolarize and then repolarize and send another action potential. In order for neurons to connect with each other and to be able to pass messages from one another, we have what's called synaptic transmission. You can see this up here where one neuron can attach to another neuron, and usually these are the interneurons where the information is processed or incoming information is integrated. And the way that this occurs, anytime two neurons attach to each other, they attach at what we call a synapse. And you can see the first neuron here, we call this the presynaptic neuron. The impulse comes all the way to the end, to the synaptic knob, where, again, we have exocytosis. We have vesicles full of neurotransmitter waiting for the impulse to reach here before they exocytose and release their neurotransmitter into the synapse. Now, the synapse is the small area between one neuron and the second. The second neuron is called the postsynaptic neuron. And the neurotransmitter will then bind to the receptors. You can see the exocytosis here. It binds to the receptors, and this then triggers the second neuron to begin an action potential. Now, a synapse, as I mentioned, it's a junction between one neuron and another one, and it also can be between a cell, an effector or an interneuron or another neuron. Now, there are two different types of synapses. There's excitatory and inhibitory. And an excitatory synapse is one that allows the sodium channels to open, and it increases the likelihood of an action potential. It kind of raises the resting potential to closer to threshold, so only a small stimulus is needed to send an action potential. An inhibitory synapse is one that hyperpolarizes the postsynaptic cell, the postsynaptic neuron, the postsynaptic cell, even if it is a muscle cell, and decreases the likelihood or the ease with which an action potential can occur. So if you think about it, I'm going to come back here so I can show you this. So here, an excitatory synapse would raise the resting potential so it's just below threshold. An inhibitory would increase the resting potential, hyperpolarize it, so that an action potential, it needs this much more of a difference in order for the action potential to occur. Okay, now back to synaptic transmission. Summation is what happens when a neuron has a number of different neurons attached to it, and this is very common. The reason why we have this is so that we can have combined excitatory and inhibitory effects at any given moment. And this allows for fine control over neuronal responses, as well as fine control of your organ systems. So for example, we have both excitatory and inhibitory on all of our organs in order to increase output, decrease output, or basically if you think about music, there's a gain and decrease. So you can either increase the volume or decrease the volume, increase the function, decrease the function, based on which neurons are triggered. Is it the inhibitory or the excitatory? Are we going to make it easier to send an action potential, or are we going to make it more difficult? Neurotransmitters, there's a number of them. They are associated with different behavioral systems. So norepinephrine really helps regulate mood, hunger, thirst, and sex drive. These are all the basal level drives. Serotonin helps promote a feeling of well-being, which is why typically when people are suffering from anxiety and depression, they'll get a selective serotonin reuptake inhibitor, where it allows the serotonin to stay in the synapse in the brain for longer. So that way, the serotonin can then continue to send the signal of well-being. And dopamine helps regulate emotions as well as complex movement. Dopamine is necessary in the cerebellum. The cerebellum, we'll talk about in a few more lectures, helps coordinate activities. And dopamine helps regulate that as well. Now, there's changes in neurotransmitters that can cause diseases. Alzheimer's is associated with decreased levels of acetylcholine. Acetylcholine is the typical neurotransmitter that moves between one and the other, so it just doesn't move as quickly. Clinical depression is decreased levels of serotonin, dopamine, and norepinephrine. Parkinson's is also decreased levels of dopamine. Now, this is it. You should be able to describe the structure of the neuron and relate it to its functions, list the different types of neurons, understand the importance of the myelin sheath and saltatory conduction, describe the all or nothing principle, explain synaptic transmission and the neurotransmitters involved, and then analyze the importance in summation in the nervous system. Thanks for joining me. I'll see you next time.